Good evening, everyone, and welcome to the June 2021 edition of the Atom live streaming events. Thank you for joining us this evening. I realized that this month's event was quite hurried and wasn't as well advertised as our other events, but we wanted to try and make sure you got your fill of science this month. I hope that you all managed to get down to the marketplace at the weekend and join in with the Atom Festival Sci Cash Challenge, and hopefully some of you won some lovely prizes. And then tonight we get to hear a talk from a brilliant scientist, so I think a good June all round. And of course, happy Pride Month as well. Hope you're all managing to find a way to celebrate that as well. So yes, this evening we will be hearing from Dr. Rebecca Smith and her talk, DNA Repair in Focus. Before we get to the main event, however, it is with great pleasure that I can announce that COVID restrictions, provided they ease when they are intended to, we will be returning to face-to-face talks in July to hear for our fantastic event that we have planned from Frank Close on the 22nd of July. This falls after the new intended date that we will have restrictions easing, and so we hope that there isn't another extension. We have spoken to the people who run the North Court at the Abingdon Football Club, and they are happy for us to go ahead. So there will be more information about that coming soon. We've got a committee meeting very soon to discuss the nuts and bolts. We're really excited about it. I'm sure you guys are really excited about it. But we are aware that some people may still not be comfortable coming to group events, may not have come out into public very much yet. And so we are looking to see how we will be able to provide this online as well. It's unlikely to be live, and we will be prioritizing the in-person event, but we will try our very hardest to make sure that you can still access the Atom Society no matter where you are and what you're doing that week. All right, so great news, really happy about it, and fingers crossed it all goes ahead nice and smoothly. Before we move on to the talk, we obviously have our science snippets. So if any of you out there are listening and you have some science snippets, please do pop them in the chat. I am being waved at by James, so I will bring him in now. Good evening, James.
Hello, good evening, and good evening, everybody. And I'm delighted with the news that we've just heard that we'll be meeting up together in the hall in July. I've been looking forward to this for a long time. It's about time. So that's a great piece of news. Thank you. I've got a couple of snippets today. The first one is a bit of a puzzle for me, really. Concerns some research recently done at the Georgia Institute of Technology by Dr. Andrew Schultz. He, no doubt assisted by some of his staff, managed to persuade an elephant to stand in a tank of water, fairly shallow water, I understand, and the elephant was then encouraged to drink this water. And then the elephant was asked to leave the tank, and they then measured the volume remaining. So they were therefore able to compute how much water the elephant had drunk. And they then wanted to calculate the rate at which it had drunk the water. And they produced the result, and the result is what puzzles me. The result they quoted was the elephant drank water at the rate of 150 meters per second air flow equivalent. Air flow, not fluid flow. So how fast did the elephant drink the water when we were given her airflow? Now, had it drunk at 150 meters a second, it'd be dead by now, I'm sure. Anyway, I don't know. So if you know the answer, how to convert airflow meters per second into the rate at which the elephant sucked the water up its trunk, I'd be more than grateful. The second
postcard.
Yeah, on the postcard. The second cryptid snippet I have today is about the country from where our speaker was studied tonight, New Zealand, the University of Otago in New Zealand, where three academics, Professors Leanne Parkin, Patricia Priest, and Dr. Sheila Williams, carried out some research. And they found that you are 5 times less likely to slip on an icy pavement if you wear your socks outside your boots. Now, I think that's very useful because, as we know, local authorities in the UK are a bit short of resources, and so we're probably going to have some icy pavements come this winter. And so if people wear their socks outside their boots, they could save themselves quite a few damaged limbs. I'm going to give it a test at the Oxford Ice Rink this weekend to see whether I can confirm the research. But I thought that was a great contribution to the studies of humankind. So thank you very much. That's it. There you go. Those are my 2 snippets.
Fabulous. What do you think would look crazier, James? Someone walking down the street with their socks on over their boots, or someone walking down the street with a canister of table salt in their hand, shaking it in front of them?
Oh, well, I'm going to try both, actually.
You're going to try both?
Yes, I've got some thick socks that I put aside for this purpose in the winter and for the weekend at Oxford Rink as well. So I'll let you know how it goes.
Okay, good. Look forward to hearing your results. Thank you very much.
I'll let you know.
I'm not seeing any coming through on the chat at the moment. I know that our speaker had a little one, but I'm sure she can bring that in before she begins her talk. For me, in lieu of science snippets this month, I hope you will indulge me a little. Oh no, hang on. There's one coming through from Michael. Bear with me. Okay, so Michael has told us that there was an announcement tonight that the Diamond Light Source have discovered an enzyme that will digest plastic, awaiting further details with interest. So I know there are already some bacteria that we know of that can do this. Perhaps this enzyme has been identified from them, or perhaps it's entirely new. But that's very exciting. Very interesting news. Thank you very much, Michael. And yeah, so what I was going to say was in order to recognize this month of Pride Month, I wanted to draw your attention to organizations that are attempting to improve the lives of LGBTQ plus individuals in STEM. We're pretty bad in STEM in terms of representation, and so there are some various groups that are working to improve that. And so if you have a moment this weekend, perhaps, I would encourage you to look up an organization called LGBTQ STEM, which was founded in the United Kingdom and hosts seminars once a year that are inclusive spaces to encourage LGBTQ plus members of the STEM community to feel open and free in discussing their science and their research without fear of persecution. There is also House of STEM, which is a community-led initiative to improve LGBTQ plus support, visibility and representation in STEM in Ireland. And 500 Queer Scientists, which is a really fascinating website that I would encourage you to go and take a look at, which is a visibility campaign showcasing LGBTQ plus people and their allies working in STEM and STEM supporting jobs. So yes, I encourage you to go and check those organizations out and enrich your lives. Okay, so without further ado, I would like to introduce this evening's speaker. She is a fantastically intelligent scientist and very close personal friend of mine. So I'm really excited to hear her talk this evening and have not loaded any pressure on her at all with that introduction. Yes, Rebecca completed her BSc and her PhD in biochemistry over in New Zealand before she moved to Europe. Spent 3 years undertaking postdoctoral studies in Munich and has since moved to the University of Rennes to continue those postdoctoral investigations, looking at DNA repair processes with a variety of advanced fluorescence microscopy techniques, including lasers, which I think is incredibly cool. She currently is at the University de Rennes in France, where her research currently focuses on understanding the molecular mechanisms of one of the earliest steps of DNA repair and how modulating this pathway can alter the efficacy of chemotherapeutic therapies. And yes, so I'm very interested. I've already seen little snippets of the talk. some very cool things going on. And so with that, I will bring Rebecca in. Hello, good evening.
Hello,
and,
good evening.
Take it away.
Oh, hi, thanks. So just my little snippet that I had heard or I'd seen earlier in this week was that the Delta version of the COVID was 90%, or the Pfizer and AstraZeneca vaccines were 90% efficient against this Delta variant of the COVID. So that was another nice piece of news for us this week. I thank you for giving me this opportunity to speak with you today. So I just wanted to share with you a little bit about what we do in our lab. So like Danielle said, we study DNA repair processes, and our work primarily uses fluorescence microscopy techniques. But just to start, I think it's important for us to just remember what is really happening in our cells, where the DNA is, and why it's important for us to repair it. So one of the ways I like to talk about DNA organization is through something I call the library analogy. So if we consider the nucleus, so this is a nucleus from a human cell, and in this small compartment, there is the entire human genome packaged into a very small area. And within this, it contains all the information you need to express multiple proteins or to regulate different processes. And so I imagine this similar to a library. So in this library, there's a multitude of different resources and books available to help to run a multitude of different processes. So in our nucleus, we can consider that there are multiple chromosomes, and on these chromosomes, there are a number of individual genes. So this is similar to saying that in a library, there's a number of different books, and say this book is actually a recipe book, then each gene is analogous to a different recipe, so it codes for something different. And then going further than this, we want to talk about how we can take this recipe or this gene and actually make a product. So our DNA is actually really precious to us. We want to maintain it in the best form we can. We don't want to damage it. So if we were to just make this product directly from the DNA, there's an increased chance that we could damage it. So what we do is basically we make a photocopy. We take our recipe and we photocopy it. And this is what we call mRNA. And then from this mRNA, our cells can make the product. So we can make this thing a protein, for example, or a cake. That'd be nice. But there are times that things go wrong. So what happens if we damage the book, for example? So in a library, a librarian would be able to say that oh, look, here is a damaged book. And she could take this book to someone to be able to repair it. And this is exactly what happens in our cells as well. So if we have our DNA and it gets damaged, we have signaling proteins, or these signaling molecules, that can recognize this DNA break, and it sends a signal to the cell to say, there is a break here and you need to repair it. And this promotes the recruitment or the accumulation of repair factors that help us repair the damage. But things can go wrong, and this is inevitable. So if this damage is not repaired properly, say, for example, this piece of blue DNA is put together with this black DNA, we can make mistakes, or what is commonly known as mutations. And if we get enough of these in our cells, we can actually develop cancer. So what we study in our lab is we study this repair process. We want to know how it works and how we could possibly modulate it to make it work better, or if we could modulate it for different types of chemotherapies to actually treat different types of cancers. So in our lab, what we like to do is use a variety of microscopy techniques to study DNA repair. But if we look at cells using white light, so just the white light that is in your light bulbs, for example, you'll be able to see something that looks similar to this. So there's a couple of things that you can see. you can see that there are 6 cells here. So each of these circles here are actually a nucleus. And surrounding the nucleus is the cytoplasm. So these lines that you can see for here mark the different type or different cells. You can also see that within the nucleus there are some darker regions, and this could be nucleoli, which is just another compartment within the nucleus itself. And there's other darker regions that could be different types of things in the cells itself. But to study a single protein in something like this is very difficult. You cannot see these proteins because they're translucent in this situation. So we need help to be able to see them. And this came in the 1960s from this amazing Japanese scientist who was interested in understanding what made jellyfish fluoresce. And what he was able to do was identify a protein called green fluorescent protein, which would emit light in the green spectra. And for this, he won a Nobel Prize in chemistry in 2008. So further in the 1990s, what was happening was basically a molecular biology revolution, you could say. And we learned so much more about how to manipulate DNA. And so what scientists were able to do was to take the DNA sequence for this GFP protein, this green fluorescent protein, and attach it to the DNA sequence of our favorite protein, for example. And with it combined together, when they put this DNA into cells, what you could see is that a green signal representing where your favorite protein was. So this was actually, you have a sort of chimeric protein with a GFP and your protein of interest. So I just want to show you 2 examples of what we can see with this green fluorescent protein. So here, what we do is we tag a protein. This is what we call when we make this fusion, is we tag a protein with GFP. And you can see in this instance here, I have a GFP tagged protein, and it expresses in both the nucleus. So this is this brighter region here. This is the nucleus. And it also can be found here in the cytoplasm, so in the rest of the cell. Compared to if you look at this protein here, so if we look at our GFP tagged orange protein, you can see that it's actually localized in the nucleus itself. And if you compare the staining, so if you look closely at this structure here in the nucleus, you can see that it looks very different to this one here on the left. So this is nice and smooth, and this one is quite mottled. And it tells us some things about cells. But just by looking at a single picture, just like this, we can't really tell a lot. So what we like to do in our lab is we like to use time-lapse imaging. So this is when we take a multitude of images over a certain time, and we can look at how the fluorescence protein changes. And also what we do is we couple this to something called laser microirradiation. So this is basically where we take a laser and we shine it down our cells to induce DNA damage. So what I have here is actually a nucleus expressing a GFP tag protein. And when I push play, you'll be able to see some really cool stuff. So basically here, I induce DNA damage in a line, and you can see that the protein enriches or there's more of it coming at the line, and eventually it goes away. And so if you look at this graph here, for example, you can see that there's a peak where immediately after damage where this protein comes, and then over time it goes away. So let's just watch the video again, because we can or can't. So here it is again. So if you irradiate, and then you can see that if there's more protein coming, and then it goes away. Okay. So this is one of the most important techniques that we use in our lab, and I now would like to just switch to giving you a bit more details into what we do specifically in our lab. So like I said before, we study DNA repair processes, but we focus on a particular protein known as PARP1, or it's actually called Polyadp ribose polymerase 1, but that's maybe a little bit too long to say each time. So for the remainder of my talk, you're going to see some images like this. So this is just a schematic that tells you, or to show you what's, it's just a schematic of DNA. So in fact, the DNA here is brown, and it's wrapped around these things called nucleosomes. And nucleosomes are just proteins that help package our DNA into this really tight nucleus. So if you are aware, there's approximately 2 meters worth of DNA in every single nucleus, but your nucleus is maybe 10 micrometers in diameter. So we need to be able to package this large amount of genetic material into a really small space. So this nucleosome is just what helps package the DNA. Okay, so I want to talk about this polyadp ribose polymerase 1. It is possibly my favorite protein, and I'm not ashamed to say it. So PARP1 is, I think, the earliest actor in the DNA damage response, in one of the DNA damage responses. So it recruits very rapidly to sites of damage. And in fact, if you remember this video, this is actually GFP PARP1 coming to sites of damage. So PARP1 is able to recognize when there is a break. It actually recognizes the break specifically, and it recruits to these break sites. And once it's recruited there, it polymerizes, or it adds a number of these molecules called polyadp ribose. It adds it to proteins in around the break sites. And this is an important signaling step, because these PAR chains can act as a signal to recruit other proteins to it, but it also has another function, and it's actually to facilitate the release of this protein. And this release of PARP1 is actually very important, because PARP1 itself cannot physically repair DNA. All it can do is say, hey, there's a break here. Someone can please come and deal with it. So that's all PARP1 does. So it recognizes a break, it adds these chains, and then other things come on. And if it puts this chain on it itself, this helps remove it from sites of damage, allowing these other proteins to come. Okay. How do we know this? So one of the ways we know this is through the use of these inhibitors. So there's a number of PARP inhibitors that are available in the scientific field, but also in the medical field, which is what I'll get into a little bit later. So these PARP inhibitors bind to PARP1 itself, and what they do is they don't prevent PARP1 from recruiting to sites of damage. So you can see here, this is before damage. Our GFP tag PARP1 is really nice through the nucleus. And then after damage, you can see it recruit to this laser line that I've cut in the middle of my cells. But unlike the other situation where you could see PARP1 being released, here you do not see PARP1 being released. It stays at the break. So if I show you a video here where I compare on this side, this is an untreated cell, and on this side it's an inhibited cell, you can see over time that in this situation on the left, PARP1 is removed from the sites of damage, but here on the right, it actually stays for longer. And this could be quite a dangerous thing for our cells. So PARP1, as I said, is actually just a signaling molecule. It says, here's the damage, and then promotes the recruitment of these downstream repair factors that actually promote or actually do the DNA repair. So if PARP1 is not removed from damage, what happens is that the break remains. And if there is if these DNA breaks stay in the cells, eventually it will lead to cell death, which is obviously not what we want. Also, it could lead to an accumulation of these mutations, and then it's also not good for your cell. Okay, so this is obviously something that the cells don't want to do. They don't want to have PARP1 stuck at DNA lesions. So they have developed a number of processes to help remove PARP1 from sites of damage. So I'm sure you are aware of these two genes, the BRCA genes. These are the breast cancer associated genes. And there's been a lot of talk even in the non-scientific world, where lost my train of thought. I'm sorry. So there's been a lot of attention put on these breast cancer associated genes, and you'll hear a lot of people say, "Oh, I've got the BRCA gene," and what they actually mean is that they have a mutation in these BRCA genes that makes them non-functional, and this can be really bad because, for example, if we have PARP1 stuck at the DNA lesions, the normal role of BRCA is to help remove it from these DNA lesions so that DNA can be properly repaired. However, if we're in a situation, so this is what you see on the left, in a normal situation, if PARP1 is stuck, BRCA1, BRCA2 can help remove PARP1 from DNA lesions. But if it's absent, PARP1 remains stuck at these DNA breaks, and this actually can lead to cell death. Now, this has been exploited in patients who have got BRCA1, BRCA2 mutations, so they don't have this function to repair DNA this way. So if they are treated with PARP inhibitors as part of their chemotherapy, this can actually lead to this cancer cell death, which is a good thing in this situation. So what we were interested in our lab was, could we find anything else that that would have the same effect as losing this BRCA1 and BRCA2 gene? So what we found in our lab is another protein of ours known as ALK1, which stands for amplified in liver cancer 1. If our cells lost this, similar to if they lost BRCA1 or BRCA2, they would actually, they were more sensitive to these PARP inhibitors. So what I have here is I have a number of cells. So I have, in each of these wells, I have plated the same number of cells, and then I have treated them with either a low concentration of PARP inhibitor, a medium concentration of PARP inhibitor, or a high concentration of PARP inhibitor, just to see how quickly these cells die. So when we talk about wild type cells, this just means this is a normal situation. They have normal DNA repair, or in this situation, it means that they have ALK1 present. And in these cells, the ALK1 knockout cells, this means they are devoid of ALK1. So in these cells, what you can see was that when we increase the treatment of PARP inhibitor, these cells actually die more than their wild type counterparts. And you can also see this here. So if we increase the concentration of the drugs, our cell survival decreases. So what we wanted to know was, also what I should say is, what we wanted to know in our lab was how ALK1 helps remove PARP1, or if it removes PARP1. So ALK1 is a protein that recognizes these chains of Polyadp ribose, or these PAR chains that PARP1 adds onto itself after binding to DNA. And what we were curious was does ALK1, after it binds to this protein, actually help remove PARP1 from sites of damage? So what we did was we took our GFP tagged PARP1 protein and we put it into either wild type cells, so these were ALK1, they had ALK1, or we put them into cells that were devoid of ALK1. And we looked at how quickly we could remove PARP1 from DNA lesions. And we did this in the absence of PARP inhibitor, or we did it in the presence of PARP inhibitor. And in both situations, what you can see is that in the presence of ALK1, so this black line, PARP1 is really removed quite efficiently from DNA. While in the blue line, which is our ALK1 knockout cells, part 1 is released a bit slower. And this is even more apparent if we add PARP inhibitor. So if we trap PARP1 at these DNA lesions, then PARP1 is released even less when there is no ALK1. So what we were able to show in this paper was that this ALK1 can actually modulate this PARP1 trapping effect. So if you have, this is what I was trying to say before, if you have ALK1, it is able to help remove PARP1 from DNA lesions, which leads to active repair, and you can end up with a good DNA situation and happy cells. However, in the absence of ALK1, PARP1 remains trapped at these DNA lesions, and this ultimately doesn't allow the repair of DNA and can lead to cell death. And I just realized that that's actually a very short talk, because that's unfortunately all I have to tell you about today. But these are the people who are really involved in our study. We work closely with a group in Hungary, and as well as someone from Munich. And we work closely with this imaging facility here in Rennes. So thank you for listening to my talk. And if you have any questions, I'm happy to answer.
Thank you very much. That was very interesting. And I especially enjoyed the videos with the lasers. I'm sure there will be many questions. Committee members that have joined, if you have questions, please wave at me and I will bring you in to ask them. I guess the obvious question for me is what's next? So you said you wanted to identify mechanisms that could be exploited similarly to the BRCA1, BRCA2 situation, and you found ALK1. What's next? Do you have any other proteins in your sights?
We do. So there are. So I should say, when I did this study, we started from a situation where we did generally a screen. So we screened for a lot of protein. We screened over the whole genome, I should say. And at the moment, we're trying to figure out which ones would be the most interesting to look at. So I have some candidates. I don't really want to say much about them because I don't have much information about them yet. But it's something that I am working on. But
so
also, yeah, many. I think what's interesting is that at the same time as that we identified this protein, about 4 other groups also identified this protein. And so this one is quite a good candidate for actually improving chemotherapy in this situation. I think maybe what is even more interesting about this guy is that sometimes, have you heard of people having developing resistance to their chemotherapy, for example? And they want to understand the mechanisms which drive this chemotherapeutic resistance. Yeah. And in the name of this protein, this amplified and liver cancer one, what you actually find generally in cancers is that there's more of this protein. So if there's more of this protein, what it could actually do is, in a situation where you don't have BRCA1, BRCA2, it could actually remove PARP1 and bring your cells back to a living situation instead of having this dying situation. So I think that is something that maybe we should focus on as well.
Cool. Just looking out for questions, committee members. Do you want to ask anything? Oh, yeah. James, is that you waving?
Yes.
Yeah.
Yes, thanks. I have a question. I'm interested in the way in which a break in the cell or in the DNA molecule, how is it detected? I mean, you said it's detected and then it's repaired or attracts repairing proteins, but how is it detected in the first place? What actually happens?
So if we look at this situation, so you can see that this protein is throughout the nucleus. There's a lot of this protein, it's throughout the nucleus all the time. And when there's a break, the protein itself has some domains that can bind specifically to the break. So PARP1, when it has a break, it has DNA ends, and the protein itself is more favorable to binding this than to not binding it. So as soon as there's something for it to bind, then it will bind there, if that makes sense.
Okay, I see, yeah.
So the break itself promotes this guy to come. It's more interesting for him to bind to the break than to just float around in the nucleus.
Okay, so you look for that protein as an indicator that there is a break and the protein is attracted to that break.
Exactly.
Yeah. Okay, thanks.
Okay.
Thanks.
Thank you. We have a question from Jane, who asks, and she does correct the typo in her next comment, but could attaching GFP to a protein interfere with its function?
Yes, it can, and this is something we need to be very careful about. So in particular, when I work with this PARP1, what we have found really recently, and which is what we're actually working on now, is if you put it on one end, the protein works properly, but if you put it on the other end, you actually interfere with it binding to an interacting partner. So this is something you need to be aware of. And so generally, we back up our studies by either looking with non-tagged proteins, so just trying to express the protein as it is, or you do it you do the inverse, so you remove the protein and do you see the opposite effect? So we do this kind of studies as well. So you're entirely right, yeah.
Yeah, it was a really good question. Thank you, Jane, and thank you,
Yeah,
Alex, for your response. Do we have any more coming through? Oh, I think there's one just about to pop up. Give us a few seconds. So Bob asks, how do you tag the protein with the fluorescent marker, and how do you stop it tagging other stuff too?
Okay, so it goes back to basically this scenario here. So to get the protein, you need to first have DNA. And what we do is we make a DNA sequence where you have the GFP sequence, and then the protein of interest sequence directly after it. So normally when you finish expressing, or normally at the end of like the sequence for a protein, there's a signal to say stop. So do not put any more stuff on here. I'm finished being made. So in this situation, the GFP would have a stop, and then your protein of interest would have a stop. But what we do is we remove the stop from the GFP, so it just continues on to make the whole protein. And so in this situation, only your protein of interest will be GFP tagged. So there's only the sequence to make this chimera protein, for example.
Yeah. So it's not that you're sticking the GFP onto the protein after the proteins have been made, it's actually in the synthesis process.
Yeah, you're making the protein, you're making the GFP with your favorite protein.
Cool. Thank you very much. Okay, I'm just keeping an eye out. We have a little bit of a lag, so I'm just checking that we don't have any more questions coming through. It doesn't look like we have any more. Oh, wait. Aha. So Susan from our committee has asked, it's not on the YouTube channel, so I can't bring it on the screen, but she's asked, what happens if there is a problem with PARP1, since it is relied on to announce that the cell needs repair, i.e. PARP1 is damaged itself?
You're very true. So I think this is the thing about DNA repair, is it's so important that there's so many redundant processes. So there's PARP1, and he's the major factor in my favorite type of repair. But there's also PARP2. So it's similar to PARP1. It does the same effect, but not as efficiently. So there's several PARPs that do the same. For different types of breaks, there's different proteins that can signal. So one of the girls in my lab works on an oxidative damage. So when your DNA gets oxidized, it produces a marker that's recognized by another protein called OGG1, or I don't remember its full name. But this guy, so yeah, basically you have multiple different types of proteins so that you can try to repair as well as you can.
Yeah.
Oh yeah, backups. That's what they are, backups. Thanks, Danielle.
Thank you very much. Okay, just while I give people a few more seconds, just if they have any more questions to ask. James, you will be interested to hear that we have an answer regarding the elephant water ingestion. So we have had someone come back. So Michael has said that according to the Georgia College of Engineering, the elephant sucks 3 liters of water per second.
It's a lot of water.
Which seems like a lot.
It does indeed, yeah.
It seems like a lot, James, but they are big animals. The trunks are very, very wide.
Well, thank you,
Maybe
Mike.
they were very thirsty. There we go. Okay. I think that's all the questions that we have for Rebecca for this evening. So it just remains for me to thank you once again for coming online. You're obviously an hour ahead as well. And I know you stayed at work to give this presentation. So thank you so much for doing that for us and giving us your time. And yes, I look forward to seeing everyone else's faces next month, because you have spent a year and a half looking at mine. And now I want to see all your lovely faces with drinks in your hands, ready to hear from Frank next month. Look out for emails with more details to follow. Cool. Have a good night, everyone.